Hey, I'm Victoria.
And I'm Tyler.
We're here to teach you about mitosis and meiosis. These are both different ways that cells replicate and divide throughout all living things. Components of the cell cycle are interphase, mitosis, and cytokinesis. We will start by talking about mitosis and how the process works.
However, before mitosis begins, it has already gone through interphase, which means it has gone through G1 when the cell prepares for DNA replication, S when the DNA replicates, and G2 when the cell prepares for the mitotic phase.
Mitosis is made up of 4 phases, the first being prophase. As the cell progresses from interphase to prophase, the chromatin in the nucleus begins to condense. The nucleolus will break down and eventually disappear. The centrioles begin to move toward opposite poles and microtubules extend from the centromeres. By the time the cell is ending prophase, the mitotic spindle starts to appear.
Following prophase is metaphase. In metaphase, the mitotic spindle completes itself and the chromosomes align along the middle of the cell nucleus, the metaphase plate. The chromosomes are organized in a circular position to ensure that the next phase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome. A spindle microtubule attaches to each chromosome, which is double-stranded.
The next step along this path of mitotic cell division is anaphase. By now the chromosomes are lined up and ready to be pulled apart. When the microtubules pull chromosomes apart, the chromosomes separate the kinetochore. This process leads to doubling of chromosomes as there were during metaphase. The microtubules are attached to each of the sister chromatids at the centromeres and pull them towards the poles of the cells with arms of the chromosomes trailing behind.
The final phase of mitosis includes both telophase and cytokinesis. At this point, the chromosomes gather at the poles of the cell and the nuclei of the daughter cells begin to form. Once packaged inside the nuclei, the chromosomes loosen. The spindle microtubules disperse and cytokinesis begins. Cytokinesis is the division of cytoplasm. This results in 2 daughter cells.
Now that mitosis is complete, the 2 daughter cells are genetically identical to the mother cell, each containing the same amount of genetic material as their mother cell too.
Now onto meiosis. It's a blast!
Meiosis is divided into 2 different sections. Meiosis 1 separates homologous chromosomes, and meiosis 2 separates sister chromatids.
The first phase of meiosis 1 is prophase 1. During this step, the chromosomes condense, and homologous chromosomes pair to form synapses, a step unique to meiosis. The paired chromosomes and the formation of chiasmata are caused by genetic recombination. Also, the nuclear envelope breaks down and the centrosomes move to the poles, just as in mitosis. Microtubules from the centrosomes attach to the kinetochores of the chromosomes and move them toward the metaphase plate.
Following prophase 1 is metaphase 1. The chromosomes align along the metaphase plate with 1 chromosome in each pair facing each pole. The microtubules from each pole attach to the kinetochores of the chromosomes that are facing that respective pole.
In the next stage, anaphase 1, the microtubules pull each pair of homologous chromosomes apart, while at the same time elongating the cell. Sister chromatids remain attached, causing chromatids to move as a unit toward the same pole.
The final stage of meiosis 1 is telophase 1 cytokinesis. During this phase, 2 haploid cells form with 23 chromosomes, which consist of 2 chromatids each. At this point, a nuclear envelope may form, or the cell may quickly progress to meiosis 2. Either way, the cytoplasm is split and 2 complete daughter cells are formed by cytokinesis. Every step of meiosis 2 occurs to each of these daughter cells individually.
Moving on to meiosis 2.
Much of meiosis 2 is similar to meiosis 1. We will focus on the major differences as well as repeat key points of meiosis 1.
Entering into prophase 2, if the nuclear envelope rebuilds itself, it gets broken down again. The spindle apparatus then forms, and the chromosomes begin migrating to the metaphase plate.
The next step is metaphase 2. Similar to the previous metaphase described, chromosomes are positioned on the metaphase plate. The kinetochores of the sister chromatids point toward opposite poles and microtubules from respective poles attached.
Moving on to anaphase 2. The breakdown of proteins holding the sister chromatids together at the centrosome allow the microtubules to move the chromatids toward opposite poles.
To finish up meiosis, telophase 2 and cytokinesis occur. Once again, 2 nuclei form, each around the chromosomes at corresponding poles, and the chromosomes loosen up. The cell pinches itself off, creating 2 separate cells. The
process of meiosis results in 4 haploid cells containing unreplicated chromosomes, allowing for genetic differences from generation to generation. These 4 haploid cells contain only half of the genetic info that their mother cell did.
Now that you understand both of these processes, the major points we want you to walk away with are the differences between mitosis and meiosis. Mitosis creates 2 genetically identical daughter cells, and meiosis creates 4 genetically unique haploid cells.
We hope you enjoyed learning about cell division and reproduction. Mitosis and meiosis!